Okay, so as mentioned already, one of the approaches where you could also focus on is going to an allogenic approach. And from an industrial perspective, but also from a patient use, they have a series of advantages. You can do large-scale manufacturing. Your product consistency is much better under control. Your cost of goods due to the scale-up is better. And what is most important for urgent situations, you can have it ready at the bedside when you need it. When you take an autologous product, it has to go to the manufacturing facility and you can see it back 2 months later eventually. So there is definitely a place for allogeneic cell therapy products. And I think there are more than hundreds in clinical trials, including also in Horizon 2020. Now, you should imagine this is like a cell transplant, what you're doing. And for people in the transplant body, you're coming with a cell from somebody else into another person's. And this can provoke, even if stem cells, especially stem cells, have a low immunogenicity profile, it will provoke immune responses. And this bare fact is a very important one from also a regulatory point of view and also from a patient point of view, because these immune responses which take place can have an impact on the safety and can have an impact on the efficacy of your final product. And this is something we really need to know. And today, there is some scarce, let's say, knowledge around that. Most comes from the transplantation, but this is very focused. Here we're working with different cell types coming from different sources, different origin, different manipulations. So the know-how about what is the immune response for given cell therapies is not known. So what would be the scope of such a program? One, of course, is to look to the different types of allogeneic cell sources. And we focus here on allogeneic, but I could say we can do it also on autologous ones, because a cell which has been cultured for some weeks in a culture disk is maybe not so autologous anymore as it would think, and could also have an impact on immunogenicity. But here we focus for the time being on allogeneic. Another thing to take into account is the disease. We will put these cells, what origin they also have, in a diseased body. So there is an ongoing pathological process ongoing, and often it is an inflammatory process. This can influence the immunogenicity and also the outcome of the potential immune response. Of course, and people have worked in vaccines, know that the route and the schedule of giving antigens is an important factor on defining what will be your immune response. Will it go there? Will it be persistent, and so on? I think all this is the basic immunology around looking to the immunogenicity of allogeneic cells, but the goal is really to do this in a clinical setting, because you can only mimic this complexity in a clinical setting, and also there you will have the possibility to look to the safety aspects as well as the efficacy aspects of such approaches. The green is not working. Okay, can I have the next slide, please? Okay. I have to go one back. Okay, good. So what can be the logical expected deliverables, of course, is to understand more in detail the possible immune mechanisms which might play against cell types. Could be of the innate nature and adaptive one. What will be important, of course, is what is the intensity of it? What are the kinetics, the specificity, persistence? Here we are in a kind of pharmacodynamics, pharmacokinetics, but more on the safety side from the regulatory point of view. What will also be important is the memory responses, because for certain diseases, patients will definitely get several administrations. So you can build up the immunogenicity. Is this a problem? Or is this not really mattering? A very important question. As I said, the underlying pathology can influence it. And then, of course, as I said, clinical safety data, clinical efficacy data, and this points to the long-term efficacy Carol was talking about. And then from a practical point of view, because a lot will be driven, like in transplantation, by the HLA matching, which might give us lessons about are there preferred sources of cell therapy products with a certain class of HLA, which would be a favorable type of product, and not working on products which are highly immunogenic. And if we do repeated use, would it be necessary to use other sources of cells so that you avoid the HLA matching or the immune responses against the HLA. And as said, it was mostly focused on allogeneic, but a lot of these principles can, of course, also be applied to other type of cell therapies in the broader sense. And as you see, the importance, of course, is the collaboration, because there's a lot of academic know-how in the immunology. There is the need to have access to clinical data and so on and so forth. So that is really the scope of this suggestion. Thank you.
Okay, that was really quick, Wilfried. Thank you very much. So we have time for questions on immunogenicity. Yes, please.
So as we heard this morning, there are so many different types of cells, mitotic, non-mitotic, where the cells are being delivered to, that can have a huge impact on the immunogenic response. So how will you sample? Will you sample different types of cell therapy? You know, some that are mitotic, some that are post-mitotic, some that are in a retinal pigment epithelium, some that are hematopoietic stem cells. I don't know. How would you balance the vastly different types of cell therapy when you're doing your proposed project?
Indeed, that would be one of the challenges, because at the end of the day, you want to learn some general lessons which can really benefit future development, but also put the focus on what are specific safety readouts to be taken care of, what is potential influence of efficacy. So the broader, the better. I think in that context, we have been mentioning very much about registries, and you should not forget there are more than 100 of such trials ongoing. So a lot of data are already being generated, but are very scattered. Each one is doing his clinical trial and is doing his readouts in his clinical trial. But bringing this together from, let's say, existing clinical experience would be one of the major things to happen, because otherwise I'm not saying you're reinventing the wheel, but you're sitting on a huge wealth of existing data which could be also exploited and guide where you could go.